You were sitting here. Oh, I see. So it's a great pleasure. Jack Gallant from here. Here we go.
Hi. So I'm going to be talking about neurophysiology data today. In case you were worried, I was going to talk about MRI stuff. This is a last neurophysiology experiment we did in my lab in about 2000. Well, from maybe 2010 to about 2013, we collected this enormous data set, and we've been processing it ever since, and hopefully we'll have a paper out on this soon. And this is a talk all about this specific visual area. Where are we? Here, on the lateral, ventrolateral surface of the macaque brain. Well, in the macaque, it kind of runs up the whole side, but in the human, this would be the ventrolateral surface, which is called area V4. So you guys all remember that the primate visual system consists of many, many distinct visual areas. In the macaque monkey, this map, which was made in the ancient days of about the 1990s, had about 32 macaque visual areas. Depending on how you count the inferotemporal cortex, there may be as many as 40 different visual areas in the macaque. In humans, there are all of those visual areas plus a bunch of others. There's probably 50 or 60 individual visual areas in the human. And all of these do something complicated, and they're all wired together in this complicated hierarchical parallel network. And any one of these visual areas has about a 50% chance of being connected not only to every other visual area, but basically every single area in the brain. The brain is a highly interconnected, parallel, hierarchical, recurrent network. Now, I'm particularly interested in this area V4, which as you can see is sort of an intermediate area in what is known as the ventral stream. You may recall that the cartoon version of the primate visual system is that there's a dorsal stream that does sort of wear-related stuff, which is mostly about coordinate transformations related to getting the information from your retinal coordinate system down to your hand coordinates that you can use to pick up objects. That's the dorsal stream that runs up the back of the head. And then there's a ventral stream that's involved in object recognition. Of course, that's an artificial distinction because these two streams, as you can see from this map, are incredibly highly interconnected and work together all the time. But I'm interested in one particular intermediate visual area, V4. Why? It's complicated enough to be interesting, but stupid enough to be tractable. That's why. I view it as an optimization problem. What visual area can you study that is interesting, but you have a hope? So when I started this in David Van Essen's lab in the early 90s, I thought it would only take a small amount of time to solve V4, and what I discovered was it has taken a lot longer. And you may not be happy with our solution, but we'll see. Now, of course, why, just to mention quickly something you probably all know, you might ask why is this our hierarchy of visual areas? You probably are all familiar with now the work in deep networks that shows that if you build a supervised learning algorithm based on neural networks, artificial neural networks, and train it to do image classification, you end up with a deep network of, say, 5 or 7, or as many as 150, depending on how you want to build your network, different layers, and these things do a pretty good job of image classification. And if you go into the intermediate levels of these networks, you can see that the receptive fields, the tuning properties of the artificial neurons become increasingly complicated. And you can use one of these deep networks, like the CAFE network, to map onto, in this case, MRI data. This is the visual system in the back of the head, and these white things are the individual visual areas, these white lines, V1, V2, V3, V4, and so on. And it turns out there's a shift from the early visual areas representing really early levels of the features that best fit voxels in the early visual system reside at early levels of this network. And the features that best explain voxels at the highest level of the visual system respond in the higher levels of this network. So that means that V4, this intermediate visual area, probably contains neurons that have features that correspond to intermediate levels of this neural network. So one way you could try to solve V4 is do exactly the same game that we did with MRI data with neurons. I could take this cafe network and I could use the individual cells, the trained units, as features and fit them to neural data and try to solve the problem that way. That might be hard because these networks tend to be very difficult to interpret. So let me give you a little background about what we know from V4 from classical physiology. This is kind of the first sort of interesting study of V4 that didn't have to do with color. V4 was first identified as a color selective area, and later on we realized it was a pattern selective area. And this is an awesome paper that was particularly awesome because this is the paper that got me a job. We found that cells in V4 were tuned for these complicated non-Cartesian basis, like these hyperbolic basis functions and these polar basis functions. At the time we did this experiment, it was all related to Lie group theory and image transformations and all that. But these weird patterns of selectivity for polar and hyperbolic patterns probably have nothing to do with anything mathematically interesting like Lie groups. They have to do with higher order shape features being represented in this area. And if you take elements of higher order shapes, like little contour elements, you can find that V4 neurons will be tuned for little subsets of complicated contours. This is a work from Ed Connor. Here's a nice study Ed did using a genetic algorithm to try to do gradient descent down all the little shape elements you could produce and trying to find shapes that a V4 neuron preferred. So they're complicated little edge shapes. If you give a person a lesion in their V4, you find that they can discriminate luminance just fine. They can discriminate orientation just fine. They can discriminate direction just fine. There's two things they can't do. They can't discriminate the orientation of an AM border, an amplitude modulated border caused by a phase shift. And they are lousy at discriminating contour curvature. So again, it seems like V4 is representing some sort of higher order features, maybe curvature features, maybe other kinds of features. And it also has this nonlinear property that AM kind of borders seem to be maybe special. And I should mention that Kurt Baker has done some nice work over the years showing that neurons in area V2, neurons in area V1 don't respond to the, the carrier and the modulator. They don't respond to the modulator in an AM signal, but V2 neurons do. Okay, so until recently, this was the best model of V4 out there. And this model of V4 is the stupidest model you could possibly imagine. It says, okay, a V4 neuron basically has a tuning curve in the power spectral space of the images. And if I take if I measure the power spectrum of the spike triggered average of my V4 neuron, I can take a bunch of images and calculate their power spectrum and essentially dot it with the receptive field and come up with an ordering of images and preferences for this V4 neuron. And this model, which says that V4 neurons only care about the power spectrum and nothing else, will explain, I don't know, they'll have a correlation with the responses of, say, 0.2, something like that, really, really bad. So this model is basically useless. It's better than a simple and complex cell model from V1, but barely. So this is very unsatisfying. So in a long time ago now, because this person was in my lab for many years, Mike Oliver joined my lab, and he decided that we were really going to try to hit V4 as hard as we could with a huge experiment involving way more data than anybody had collected before, and then a hugely parameterized model with way more parameters than anybody had collected before. And this took Mike like 7 or 8 years to do. He did all the work in the project that I'll talk about. And now Mike did such an awesome job. He's now at the Allen Institute basically implementing deep learning models to model the mouse data that the Allen Institute is collecting, which has its own set of issues. So we had to make a choice at the beginning of this experiment. Do we want to use classic simple stimuli like my non-Cartesian gratings or Ed Connor's little shapelets? Or do we want to use natural stimuli? And of course, in my lab, we always use natural stimuli because I would rather search through the stimulus space that I know is relevant than be successful in a stimulus space that may not be relevant. So we use natural movies for this experiment. And then the problem was, how many natural movies should we use? And we took advantage of the Utah array technology. So we implant this Utah array in area V4 of a macaque. And these Utah arrays can record data for, if you're lucky, 3 or 4 years. And every day you come in and you will end up with signals on maybe about a third of these Utah arrays, and you will end up with somewhere between, say, 1 to 4 sortable spikes. Now, in a day, you might get 1,000 trials out of an animal, which might end up with giving you maybe a couple thousand seconds worth of data. That's not really much data. So if you can only record from a neuron from a single day, you wouldn't really have enough data to fit a sophisticated model. If you remember, thinking back when you took engineering at some point, you can model any system as any stationary system as a Taylor series. And if it's got a memory, you can model it as a Volterra series. And if, say, you have a fourth order system or a fifth order system, the amount of data you need to characterize these higher order nonlinearities is proportional to the nonlinearity. So as we move down the visual stream and we end up going from second order models to third order models to fourth order models, we need exponentially more data. So we have to have a way to get more data. And to do this, we developed a method that was independently discovered in Jim DiCarlo's lab at MIT, which Jim DiCarlo came up with a good name for it called the fingerprint method. The idea is you bring your animal into the lab every day and you play him this movie. This movie has 450 frames of video. And so you play this movie several times and you plot the peristimulus time histogram of the neuron to this movie. This is just the spikes over time to the movie. And now when you bring the animal back tomorrow, because the Utah array will have moved in the neuropil, the brain will have moved a little bit, some tissue may have grown. If you look at the spikes, if you look at the waveform on the oscilloscope for a neuron off your Utah array, it won't look the same and you won't know it's the same neuron. But if you then run this fingerprint method, the neuron will give you exactly the same response. And because V4 neurons are coding in a very high dimensional space, every single V4 neuron is different. And they all have their own individually unique receptive fields, and so you can perfectly identify individual neurons using this method. So we use a combination of the spike waveform and these fingerprints, and you can see that this is one neuron we recorded for 37 days. So we have single neurons that we have 3 million frames of video on. And I have a simple rule in life. If I can't figure out what a neuron does after 1 million frames of video, I don't care what that neuron does. That's my rule. So we have a lot of neurons. We have 800 neurons with on order 1 million frames of video. And that means we might really be able to do something. So you may recall, I'm sure somebody talked about this, that V1 neurons look like little Gabor filters in the classical receptive field. But as he pointed out earlier, this is a poor model of V1 because this only explains the classical receptive field. And it doesn't really explain all the non-classical surrounds. And those non-classical surrounds, as a lot of people have shown, in natural movies have huge influences on the cell's responses. So we need a model that will be able to model this and the non-classical surround. And that we need a model not only that will model V1 and its responses to natural movies, but we want to have a model that will model V2 and its response to natural movies. So Mike, after a long time, came up with the following framework, which turned out to work great and be very elegant. I have as my input a movie. And I have a bunch of V1 neurons that look like complex cells and a bunch of V1 neurons that look like simple cells. But any given V1 neuron is going to have a simple cell and or a complex cell model, or maybe a combination of the two for its classical receptive field, and a bunch of these simple complex cell models for the non-classical receptive field that reflect the input to this neuron, and they may be delayed in time. So we have a framework basically using deep networks, but they're constrained deep networks that implements this. It's called a deep time delay network. And essentially, the input to the deep time, this looks like a normal deep network, but the input is essentially a bunch of simple and complex cell transformations of the pixels that are spatially localized, and there are time delays. So the network doesn't only get this frame like a normal deep network would get, it actually gets at each time step the past history for, say, 100 milliseconds. And then the network can learn what combination of simple and complex cells it needs to, at various time delays, it needs to accurately model the cell. Now, this V1 output goes into a V2 module, and the V2 module turns out to be exactly the same as the V1 input, except scaled up. So essentially, the input to V2 is these pooling filters. These pooling filters look like complex cells and simple cells, except they're operating on the rectified output of V1. And then these are pooled and sent to the output. Okay? So this, you can think of this model as a stacked V1 model. There's a V1 model. The input goes into V1, does whatever V1 does. The output of that goes into another V1 model that only differs in terms of scale. And these parameters all fit. So this deep time delay network has a lot of parameters. There's thousands and thousands and thousands of parameters, but we have millions of frames of video. And we use very strict cross-validation where we use a huge data set to fit the model, and we use a completely separate data set of completely different natural movies to test the model for each and every individual neuron. I can talk to you more if you want to know more about how this network is designed. You can ask me. Mike is, I think, the world's expert at this point of fitting deep networks, which traditionally require infinite amounts of data, to very small amounts of data, like the responses to 1 million frames of video. That turns out to be a hard problem for deep networks, and Mike came up with a bunch of tricks to make these networks more efficient in their design, to basically lower the size of them, and to factorize various parameters in order to reduce the dimensionality of the input into the network so that it could accurately learn.
And the output is the time response of one cell?
The output is the time response of one cell. Yeah. OK, so these are the results for V1 and V2 mixed. So this horizontal axis here is the correlation, which we just used because everybody knows what the correlation is, between the predicted response and the observed response. This is the number of neurons, because this is a histogram. And the traditional models of V1 and V2, if you just have a simple and complex cell model, you'll end up with a big Gaussian blob whose mode is around, say, 0.5. If you take, say, a cafe network that was trained on natural images and use the local units of that network as parameters to fit to your neural model, you end up with this blue distribution. That network actually works pretty well. But if you use Mike's hierarchical convolutional energy model, that's what he's calling it, you end up with this green distribution. And this green distribution, although the best cells, the mode here is about 0.8, there are very, very few cells that get up to, say, 0.95. You might think, well, 0.8 isn't a very good correlation, not explaining all the variance. But we're explaining the responses of the cell in 16 millisecond time bins. We're not explaining the whole PSTH averaged over 100 milliseconds. We're explaining the highest frequency we have available in the data, which is the video frequency, 16 milliseconds. And in 16 milliseconds, a V1 through V4 neuron can give somewhere between 0 and maybe 5 spikes. So the distributions of spikes in these 16 millisecond bins are not Gaussian, they're Poisson. And if you're trying to use correlation to characterize a Poisson spiking event, then this is about, we're basically at the noise ceiling. We can't really, it wouldn't matter how much more video we collected, we can't really predict these cells any better than we are. Now, this is the comparison of our model, which is on the vertical axis, to several popular models, a neural network versus model, which is this blue thing here, the Nicole Rust's model, which is a good model, and sorry, I don't know what's going on with these little squares, and a convolutional subunit model produced recently by the Vintage. And you can see in all three of these cases, our model works better than the other model, especially when we have a lot of data. So this model is, as far as I know, the best model out there right now. Yes?
Sorry if I missed it, but the kernels in the network you're fitting, do they have a temporal component, or is they purely spatial?
They have a temporal, well, they have an implicit temporal component. So they, so basically you have the spatial component at each time lag. So it's essentially kind of like thinking like an FIR filter.
So you can weight sum over time then.
Yes, yes. So that's exact. So there's two reasons this thing works so well. One is there are thousands and thousands of parameters. Oh, what happened? It disappeared. I'm sorry, I had it on here. Oh no, it's coming up. I put it on a different slide. I'll tell you about the parameterization in just a second. Yes, somebody else has a question. Yeah.
I just want to make sure I'm clear. You're predicting single trial responses, or do you repeat the movies and try to predict the average case?
Oh, that's also a good question. So we're predicting single frame responses where the response means the average over repeats, okay? Because that's really the only thing you can predict, right? If you're trying to predict a single frame, then you're essentially trying to predict a Poisson number, and your noise ceiling will be very, very low. If you're trying to predict the average, then your noise ceiling will be higher, so that gives us more headroom, right? So on every single one of these cells, we collect a fit set where the constraint on your fit set for these kinds of problems turns out not to be repeats. It turns out to be how much you sample the parameter space. So for the fit data set, you only do one repeat, and you collect as many stimulus response pairs as you can across all the stimuli. For the fit data set, your constraint turns out to be your noise ceiling, which is how close the thing you're trying to predict is to the true population thing you should be trying to predict. We only have a sample mean. So estimating that sample, to get that sample mean good, you have to use a lot of repeats.
So the second one was the evaluation set, not the fit set, right?
Yeah, the second one was the test set. Sorry. Yeah, sorry. I use, yeah, excuse me, thank you for catching that.
And then, sorry, each of
No,
the
go
blue
ahead.
dot, each of the blue dot is a single cell response?
These are a single cell from V1, V2, yeah. So this thing works well. That's the short answer for that. Now, we want a V4 model. I mean, who cares about V1 and V2? Come on. Actually, we don't really know what V2 does, so that's interesting too. But we want a V4 model. How are we going to do that? Well, we stack. We take the V1 model, shove it into a V2 model that pools, shove it into a V4 model that pools. And in fact, you can tell where this is going. The claim of this is going to be it's V1 all the way down. It's just V1, 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 V1. You keep stacking V1 on top of each other. Eventually, you get face cells or something. So that's the idea. So we've got 3 V1s, but this one's V4. So how well does this work? Oh, here's the parameters. You can see the model. That's why I moved the parameters too. See this? These V4 cells are big, so you need 172,000 pixels as the input. You have 26,000 parameters that you're fitting. Fitting 26,000 parameters seems like a lot, but we have a lot of experience fitting these kinds of models. Now, there is one trick when you're fitting V4, which everybody in the V4 field ignores, and we did for years and years and years too.
What's convolutional energy? What's the energy part of it? Just squaring?
Because these are just an energy model. Yeah, you know, complex cells kind of an energy model. And this is implemented as a convolutional network, so it's convolutional. And then it's hierarchical, so that's his name for it right now. We call it MollyNet in the lab for Mike Oliver Net.
Convolution and then square and then convolution and square.
Yeah, essentially, right. So, so there's going to be a bunch of these units, and they all have a convolutional operation associated with them. They get half-wave rectified, right? And then, so that's a nonlinearity. And then they get fed into a bunch of other convolutions and half-wave rectifications. The main nonlinearity here is this half-wave rectification.
So the top ones have the energy in it, the lower ones not.
Yeah, but these are fit simultaneously. So the algorithm picks for a cell how many of these does it need, how many of these does it need, what their spatial frequency should be, what their orientation should be, and where they should be in the visual field. So the first thing we do is we have another convergent method that finds the rough receptive field of the cell, and we cut out an area of the video 3 times the size of the receptive field, and the network is allowed to fit anything in there that it wants. Yeah.
So in that V2 box, you're showing some oriented filters, but that's going to be learned, right?
They're learned.
But, so that's just like a stand-in for whatever it is.
Well, they have to be of this form. So it's not just learning. It's not like a deep network. You can't learn anything. It has the units have to be of this form. Right?
You just said this orientation filters of V2 that's actually the same as in V1. So that means it's hardwired. But the units that are the output on which you have these oriented things, they are learned. That's how I understood it.
There is a huge library of these functions that the system is allowed to select from.
And to combine them by learning. But the library is given.
The library is given.
So the library of oriented filters, even in V2, are given. But
yes.
They work on
it's the same one as
the combinations that V1 has produced.
Yes.
And the whole network is trained end to end with the gradient method.
Yes.
It's not layer by layer.
No, end to end. Yep, just with backprop. So these, you know that these V2 units are going to be simple and complex cells because they're constrained. That's what makes this different from a deep network, right? This is everything we know about the functional architecture of V1 is built into this network, right? Including another trick that turns out to be important. So V4 receptive fields are big. And they're so big that they actually are not circular in the visual field. And that is because, if you remember, V1 implements a log polar map. So there's a retina, and you take the log polar transform of the retina, and you end up with a huge foveal expansion and a peripheral compression. And if you imagine that this is your V1 map, and you have a cell receiving input from V1 into V4, but the receptive field, the anatomical receptive field projecting onto V1 is a circle, that means that receptive field will not be a circle in the visual field. It will be some weird egg shape. And that was a cool observation first made by Brad Motter. And so you have to include this log polar transform in the input, otherwise you don't get such good predictions. So that is actually built into the model. The log polar transform is stuck in at the very front end of this model. Okay, and this is the histogram for V4. Now, if you may remember, at V1 and V2, we were up here at the noise ceiling. V4, we're not at the noise ceiling. But if you notice, the mode here for V4 is a correlation of about 0.5. And if you remember earlier in this talk, I mentioned that the classical model of V1 predicts a correlation of about 0.5. So this V4 model works as well as the classical model of V1. And people have been saying for 30 years that we understood V1 based on the classical model of V1. So I'm going to assert we understand V4 based on this correlation coefficient. And if you don't like it, go collect 10 million frames of video, because that's what you're going to need. You might say, why don't we have better predictions here? And I will tell you, these errors are not Gaussian. Our prediction errors are not Gaussian. And that is because these prediction errors have a long tail. So what happens is most of the time these predictions are pretty accurate. But when this thing fails, it's because some specific image came in that triggered some higher order nonlinearity that we don't have in our model because we don't have enough data to fit it. And the prediction and the model go way off whack. And so we end up being way out in outer space. And that, of course, since this correlation is assuming Gaussian error and our error is not Gaussian, it looks like we have more error than we have. In general, this works pretty well. And the reason it doesn't work is because we couldn't sample the higher order nonlinearities well enough because we don't have enough data. Yes?
Are those large errors typically under predictions or over predictions?
They can be anything. Yeah.
So Jack, there's another reason the model might not work. Maybe it's a highly constrained model, right? Maybe you over-constrained it. And if you had infinite data, you would still make tiny errors.
Possibly. The reason we think it's maybe not over-constrained is that if you remember back, well, I only had it for the V1, V2. I have it for V4, but it's not in this slide deck. We have compared this constrained model to just taking any standard off-the-shelf deep network that's been trained on natural movies or natural images and using those artificial units as essentially features and finding weights and then using that network. So that network has a lot more potential support, and that network does not work very well. But that may be because it has too many parameters. So if we could take an infinitely large data set and fit it to a neural network trained on an infinitely large set of data, you're right, that might do better. OK, so what do these things look like? Really, that's what we care about. What do they look like? Can I figure out? I have some intuition that V1 implements an ugly Gabor wavelet pyramid with nonlinear interactions in the surround. What do these other things look like? Well, let's look. Here's a bunch of V1 cells, and they look like V1 cells, pretty much. This is a Gabor wavelet, Gabor wavelet, Gabor wavelet. This one, you notice, is not Gabor wavelet. It's a little more complicated. And you'll also notice there's some subtle surround going on here. And these surround weights, even though they're all low, they turn out to be absolutely important for predicting the response of the neuron. And if you don't have them, you won't be able to predict the response of this cell. So all these cells are. Is there a question?
Yeah. What are we seeing? This is just across time, the receptive field?
Yeah. These are the receptive fields over time, because these are spatiotemporal receptive fields.
Okay. Computed how?
Sorry?
Computed how? White noise?
Computed how? No. These are estimated back from the FIT model.
The linear model. I mean, you're assuming a linear model, right?
No. I think you're right. I think he fed white noise into the model. Yeah, I think so. I wouldn't guarantee it, but I think that's what he did. Yeah. Yes?
For the complex cells in V1, do you get the 90-degree shift or not?
Yeah. And in fact, some of these, you'll see it'll move. This shifts over time. I don't know if it's this one. But they'll shift. They shift both in space, and sometimes they'll get counterphase in time. They can be complicated. Qualitatively, these look kind of like simple and complex cells with surrounds. And there's nothing that this model reveals in V1 that you wouldn't have expected from a more full than classic, Campbell-Robeson view of V1. I should point out that all of these receptive fields are at the fovea. This had one cool side effect, this study. When we put our first Utah array in, we accidentally put it in the fovea. So we had foveal receptive fields. These receptive fields are like 8 pixels across, and they're within 1/2 degree of the fovea. So this is the first foveal awake monkey data that has been reported. And there's always been a long question of how much do these foveal neurons look like other neurons, kind of which was implicit in Bruno's talk. And the answer is they look like other neurons. I mean, they may, in general, right? So that's cool. Now let's go to V2. Here's the V2 neurons, and you can see these are bigger and maybe a little bit more complicated. This one maybe has some more non-classical surround. They seem, oh yeah, it can also fit color. You can see this is a color selective V1 neuron. There's some color tuning in here. They're not hugely different, and that's kind of consistent with the V2 data we have. But then we go to V4, and everything just falls apart.
Where does this very pronounced grading structure coming from?
It's all the input are complex cells and simple cells. So the features right? The basis functions that are being fit in this model are complex cells and simple cells, and so they're going to look like gratings. But you can have an arbitrarily complicated selection of gratings going into your model. And in fact, these models have somewhere between 7,000 and 20,000 parameters that are being fit, right? So there's a lot of gratings that could go in, and so you're just pulling out the most prominent ones. But remember, this model fits better than anything else. So in it's well known that V2 cells you can model as grating selective just like V1 cells, except they'll have other nonlinear properties. For example, V2 cells can be amplitude modulation selective, right? For an AM grating rather than a normal grating because of the rectification.
I still don't understand how you calculate these. I mean, you have a highly nonlinear system.
Okay. So ask me later.
Okay.
All right. I'm going to get thrown off the stage in 2 minutes here. Yeah. And this is the interesting part. Here's V4, right? These receptive fields are really ugly, hideously complicated. You could not go home and tell your kids what these receptive fields are. They're just really complicated. Every V4 cell is its own special little butterfly of hideous complexity. It makes no sense. There's all kinds of weird things in here, and they're ugly. Now, I should point out it's also difficult to figure out what these things are selective for. So one thing Mike did is he went through and he found the movie clips that we know objectively activate the largest response from V4, and then he essentially fades that movie clip into the receptive field. So you can essentially see what it is in the movie that the receptive field is sensitive to. And if you look over here, you'll see that this receptive field is responding to this weird segment of this octopus or something gill that happens to be inflating and deflating over time. And this receptive field looks nothing like the feature out of our million frames of video that most activates this receptive field. So there's no way to look at these receptive fields and figure out what they're selective for in natural images. There's no way to look at the natural image that elicited the largest response from the cell and figure out what it was in that image that would have elicited the large response. It's just complicated.
But couldn't you have your good ones that do a better job than the bad ones? They can't be all equal, can they? No,
the good ones they can be equally inscrutable is what they can be, right? So if you notice, this correlation is 0.65, and if you look at the relationship between the receptive field, which looks hideously complicated, and the image, you can, once you squint for a while, you will find some relationship, but it's not always obvious. These are complicated intermediate units that you get by pooling over successive levels. And my notion going into this, because I'm a classically trained neurophysiology guy, is there will be some representation in some basis that I can interpret. And the problem is that's not really true. All right. So you can use the receptive field complexity to, say, make a division across your array from V1 to V2. That's good. You can see these all look like V1 neurons. These all look like V2 neurons. That's great. And then you can go through, again, just like I showed you a few minutes ago, and try to predict what the best stimulus is and what the best frame is, and try to look at the correspondence here. This is the V1 and V2 inputs to this V4 neuron. This thing, like, look at this. This is who the hell wants to think about what this does? Only a mother could love this filter. This is just horrible. You're never going to figure out what this is. And you look at the stimuli that drive it, it's like, oh, God, it's just hopeless. It's just hopeless. So now, of course, if you're physiologists, think, I got to come up with cell classes. I got to put these things together somehow. That's what we do. That's what a good physiologist does. I make classes of my neurons. You can't make classes of these things. The parameter space is, there's 20,000 parameters, and I've only got 800 neurons. So I can't really do any kind of cluster analysis. It's going to mean anything. So in that case, we used the most powerful computer we had. Mike used his brain. And he squinted at these for a long time. And he decided there were 4 clusters, where clusters means I squinted. And he decided there were contour selective cells, color selective cells, color texture selective cells, small feature cells, and large texture cells. And this was like a year ago. And then he redid it more recently. And he came up with these classes, which are contour selective cells, curvature selective cells, temporal contrast cells, which you can see change over time in a sort of on-off, push-pull kind of way, and stripes. Now these stripes are an interesting class of cells because you'll notice these all look like AM filters. They all look like AM, where the carrier and the modulator are at different orientations and different frequencies and different colors. I'm done, mostly. So that's cool. Wait, what is that there? One more. Come on. Oh, come on, you beast. Well, I'll be done if I can't go to the next slide. So I should mention that you can use these. You can do an in silico experiment once you have these filters and replicate every observation that anybody's ever made in V4, which is handy. So this is not a real V4 non-Cartesian selective cell. It's a fake V4 non-Cartesian selective cell. And if you show non-Cartesian stimuli to this cell, you'll get the same tuning. So that's nice. So this accounts for classical results as well. And this relates to two interesting threads from previous research. One is this thing called the scattering transform. Some of you may know about from Stefan Malat that you can use for vision. And another is this cool paper from Karklin and Lewicki from 15 years ago, where they basically took Bruno's sparse coding model and they stacked it on top of another sparse coding model. And they ended up getting receptive field properties that look remarkably like the ones that we see in this model. And that's it. Thanks very much.
We had already a couple of questions and running late, but I think we should have a couple more.
So I just want to point out the ugly thing about this model is you can't understand it. The cool thing about the model is you can understand it. Because what you can understand is the structure, right? V1 goes into V1, goes into V1, goes into V1. That's really easy and elegant and simple because we know how to build a V1. So you know you can build that. Figuring out what it does is the hard part. Yes, Yves?
Yeah, you're using clips of movies. So what happens if you're then replaying your prediction on randomized frames?
We do have controls on the randomized frames, and if you randomize the frames, then the temporal kernel will do a really poor job of predicting the responses. What ends up happening if you randomize frames is you've taken the power spectrum, the temporal power spectrum, and you whitened it, and that tends to linearize the responses of the cell. Showing white stimuli to a nonlinear neuron tends to give you a linear response because you don't have enough nonlinear of the higher order terms to extract the nonlinear features. And so what will happen is all the nonlinear components of the temporal response will make poor predictions of that linearized data. Doris?
Like conceptually, just because you don't understand what it's doing in simple terms doesn't mean that it can't be understood in simple terms, right?
Sure. Sure. Okay. I mean, but it depends on what you want as simple, right? I don't think it's going to be understood in simple terms that we tend to think of in 1980s, neurophysiology. But the scattering transform.
Like Bartlett Mel talked about, that the cell is coding a boundary or detecting a boundary. Like I would consider like the simple function.
That I think is going to be a hard sell because a lot of these cells, I mean, they code textures and boundaries. Obviously, because that's the stuff in images. But they do it in hideously complicated ways that don't bear any systematic and simple relationship to specific textures and boundaries. Right?
Well, last question, David.
Well, I mean, what are you complaining about?
This is like another version of Doris's question. So, do you think you could add some extra bells and whistles that would force, I don't know, more sparse representations or more comprehensible representations without making the performance any worse? I mean,
no.
You don't have any proof that that this specific network is the best you can do.
No, except for this poor guy who spent 10 years of his life basically making it the best he could. No, other than that, I don't. This is the lowest parameter model we have that fits this well. So Peter also asked about this. Why don't we just use a neural network? We can train a deep network on natural images or natural movies, and then take the learned units as features and find a set of weights to map those features onto my neuron's response. And you can do that, but A, the thing actually won't work as well, at least right now, as this. And B, this is much lower dimensionality. So if we take a deep network and use it to fit our data, we need, say, 100,000 parameters, and this thing only needs 20,000 parameters. One of the nice things about this is it's a pretty low parameter model, considering what it does. So I'm not saying that there won't be any intuition, and Doris may be annoyed because, after all, V4 is dumber than IT, right? So shouldn't we be able to, if we can't understand V4, then maybe we can't understand IT, which maybe is why you asked that question. But, it could be that there are modal points in vision that are more understandable than others, right? We are animals that can pick out individual simple features. And we're animals that can pick out objects because we're designed to pick out objects. But that stuff in the middle, we don't ever really talk about that much. Nobody ever talks about like this section of my head, right? It's that these intermediate areas of weird contours and textures are not something we have any conceptual access to. So I think it's hard for us as animals that evolved to do image recognition to basically look at these data and make any sense of them.
So I realize that there are more questions pending, but I think we should move on.